Hey everyone, and welcome to our video on transmission. This is part of our Module 5 series on neuronal communication, and we're looking at specification reference 5.1.3c, the generation and transmission of nerve impulses in mammals. In our previous video, we had a look to see how an action potential is generated in the first instance. And what we're going to be doing in this video is having a look to see how that newly generated action potential then moves through the rest of the neuron. If we think back to last video, we said that this action potential is going to cause those sodium ion channels to open in the neuron. Now, as those sodium ion channels open, that means that the sodium ions can flood into the neuron via that process of diffusion, because if the channels are open, they just go on in. As a result of that, what we find is we get this increase in concentration of sodium ions inside the neuron itself, and because we're then getting that higher concentration of sodium ions just on the other side of the gate, they then diffuse down that concentration gradient and move along the axon. And basically what we're seeing here is just a movement of these charged particles, these sodium ions. So they've entered via diffusion through those now open sodium ion channels, and then they're just going to diffuse down their concentration gradient along the axon of the neuron. And this is what we term the local current. As those sodium ions are then diffusing down the axon, we're getting this slight depolarization of the membrane, because obviously we've now got a greater concentration of sodium ions in that region. So as a result of that slight depolarization of the membrane, then we get more of the voltage-gated sodium ion channels opening further down the membrane structure itself. So as a result of that, more sodium ions can enter and we get to that full depolarization. And this just continues all the way along the neuron until we get to the end. So let's have a little look at this in visual form, just for those of us that are struggling to actually see how this all ties together. Now, I do apologise, you have the grand total of my amazing paint skills. I am not an artist, this is why I teach science and not art, but hopefully we can all pick out the key things here. So, first of all, we've got the outside of the cell here and the inside of the cell up the top of our diagram. So this bit here is our cell membrane. The little blue circle down here is our little sodium ion. So what's going to happen is that when we've actually got that sodium ion channel open, these sodium ions are just going to diffuse from outside the cell into the neuron itself. And what happens is they diffuse in, and then we get this higher concentration of them building up inside the cell just on the other side of that voltage-gated channel. And because of that localized increase in the sodium ion concentration, then what we see is it's going to diffuse along the axon or the dendron, and basically it's going down that concentration gradient. Now the next thing that's going to happen is that sodium gate, which was initially closed, is then going to open because we've got that slight depolarization. So basically what we're going to see is we are going to get this action potential moving along the neuron as more sodium ions enter, and therefore they set up the next action potential as they go. Before we move on to the other method of our transmission in the neuronal system, what we're going to have a little look at is a structure, the myelin sheath. This is hopefully something we heard about back at GCSE, and you were probably told it's all about insulating the nerve cells, okay? Now, we're going to upgrade that ever so slightly because we are now A-level scientists and not GCSE, and we're going to describe the function of that myelin sheath as insulating the axon to increase the speed of action potential transmission via saltatory conduction. So that little function bit I've given you there is actually a great way to ensure you get maximum marks on any question that asks you the function of the myelin sheath. Because in fact there are typically 3 marking points within that one little sentence. So great thing to chuck on a flashcard. In terms of what the myelin sheath is, it is an insulating layer, and this is made of a fatty material from these things called Schwann cells. And these Schwann cells are very tightly wrapped around the neuron. So they're really closely associated and very tightly packed in there. Now that's obviously going to give it that insulating property because there's not those gaps that things can squeeze through. The key thing that this myelin sheath is doing then is it's preventing sodium and potassium ions from diffusing through. If we just had the actual membrane of our neuron, then we would have those channels which could allow the sodium and the potassium ions to diffuse across it. Wherever we've got this myelin sheath, they can't. So we don't have a complete myelin sheath. It actually has little gaps every so often called the nodes of Ranvier. So basically you've got the Schwann cells tightly wrapped around the neuron, little gap, node of Ranvier, another Schwann cell tightly wrapped, and so on down the length. So because the myelin sheath is actually preventing these ions from diffusing across the membrane wherever those Schwann cells are, the only place the ions can actually enter the neuron is at the nodes of Ranvier. So what we're actually seeing as a result of this myelination, the local currents that we generate through those movements of those sodium ions are going to become elongated because the sodium ions are only going to be able to diffuse from one node of Ranvier to the next. That's the only place they can actually enter. The rest of that, they've just got to diffuse along the axon or the dendron. So basically, it gives this appearance of the action potential jumping from one node to the next. And this is what we mean by saltatory conduction. They're going back into good old Latin for that, but we're not going to worry ourselves about that because it's a dead language. Now, what we actually find is that because we've got this saltatory conduction, this jumping from one node to the next, then we've got much faster transmission in these myelinated neurons. And you can see there that the speed can be up to 120 meters per second. Now we are going to take a second just to have a little look at our scientific notation for our units, just to reinforce this one because it's always a silly mock throwaway on an A-level paper. We are no longer going to be doing our little M/S's, they're banned. Whenever we're doing meters per second or whatever it may be, then we're always going to have our first unit, the second, and then we're going to use that superscript -1 to use the per basically. So that is meters per second. Remember how to use the correct notation to not lose silly marks on an A-level paper. So what does it actually look like then? Well, again, here's our lovely little diagram that I've drawn for you. We've got our little node of Ranvier just here, and these are going to be our Schwann cells of the myelin sheath. So we've got the inside in the little purpley colour and then the outside on the top of our diagram in this case. So what we actually see happening then is our little blue circles are still going to be our sodium ions. So all that happens is they are then going to be able to enter at the node of Ranvier, but then they have to just diffuse down that concentration gradient. So the only place they can actually enter is at the nodes of Ranvier. Other than that, we get that elongated local current as those ions are having to diffuse down that axon or dendron. Do remember what we said previously. There is this all or nothing rule as far as these action potentials go. They all have the same intensity, plus 40 millivolts. If an action potential is travelling, it is plus 40 millivolts. That's it. If it's not travelling, it's 0. So it's plus 40 millivolts or 0 millivolts. Now, one thing that hopefully we do know is that in terms of our understandings of different stimuli in the body, they're not all the same. We understand that there are different strengths of stimuli somehow. And the way that we can do this through this all or nothing rule is by just having different frequencies of action potentials. Because we can't make the action potential bigger because they're all plus 40 millivolts. So if you can't increase the size of it, you have to increase the frequency. So what we find is that by changing the frequency of action potentials that arrive in the brain, that's our way of interpreting the actual strength of the stimulus. The more frequent the action potential, the more intense that stimulus is. So if we actually have this higher intensity stimulus, that's just going to result in a greater number of those sodium ion gated channels opening, and therefore we're going to get more generator potentials being produced. As a result of the greater number of generator potentials, we're going to get more frequent action potentials, which then travel along that sensory neuron to the central nervous system. That more frequent arrival of action potentials tells the brain this is a stronger stimulus. Hopefully you found this video useful, and of course don't forget to subscribe to the channel so you can see the next time I upload a video, and of course head on over to the website where you can find a range of other resources to help you in your A-level studies.